First of all, my name is Andy Wyenandt. I'm the Extension Specialist in Vegetable Pathology with Rutgers University, and I'd like to welcome you all to the Garden State, Atlantic City, the great state of New Jersey, and thanks for traveling some of you have come from far distances, and it's not the easiest place to get to, but it is a fun place, so hopefully you're enjoying yourselves. We're going to try to stay on track this morning. We have a tight window for lunch, so I will be in charge of this morning's program. And as you see me creep closer to the podium this morning, that means your time is quickly running out. For those of you who haven't checked that, I think we'll have to do that somewhere over the lunch hour. I think lunch might go a little bit longer than normal, but we have some flexibility this afternoon to make up on some time because some of you are staying and need to check out like myself today. If you have any questions about anything, you can see me or Ann in back of the room. Raise your hand, Ann. There she is. She's done a tremendous amount of work to help put this program together, and we want to thank her as well. So with that, I'd like to invite Dr. Jim Simon, our team leader from Rutgers University, who put together this program along with myself and a few others from Rutgers and from other universities. And we'll go ahead and get started. Welcome, Jim.
The first presentation this morning will just be an overview of how we've come together. For many of you as growers, you might only be asking, what do I do to control my downy mildew? What do I do to control my other basal diseases? And we hope through today and actually tomorrow, a lot of those answers, not all, will hopefully become clear. And as a consortium, as a group, we'll all share ideas as to how to move forward. But to put the whole project in context, and some of you have attended last year's annual basil workshop down at Homestead. Many of you have come each year in January to hear what's going on with basil, always asking us if we have the new resistant variety or if anybody else has a new resistant variety or what's going on and what are the right chemical controls. And these are always a work in progress, but I wanted to let you know the context of what we set out to do, who's involved, and where we hope to go. And then in concert with you and with all the different researchers as a consortium, hopefully we could make progress. It is rather amazing that we came together 4 years ago when you as growers and seed companies and others were reporting to us the devastating losses from downy mildew. So many of us from both Rutgers University in concert with our partners at the University of Massachusetts. We have Robert Wick and Lee Joan Ma here, if you could raise your hands. Okay. From them, they're working on the pathology of the disease. Many of you as growers ask, well, if a chemical control works, how variable is the downy mildew? Can everybody hear me in the back? Yeah. Okay. So the question is, if it's a very homogeneous disease, can it mutate, and will the chemical control that potentially works today work somewhat effectively, or if you have good luck with your chemical control treatments, will it work 2 years, 3 years down the road? So the UMass group is really focusing on the genetics of the pathogen itself. Okay. Then in concert with those of us, our colleagues at University of Florida, the researchers include Rick Raid, Chuan Zhang, and Ann Hartman, who's down at the back, okay, and Jamil Patel. Jamil's here too as a postdoc there. They focus on the pathology. What are the chemical control agents, the organic, the non-organic control agents, and different production strategies that growers could use in lieu of there not being necessarily an improvement in the genetics of the materials. So we're delighted that University of Florida, that really was the epicenter where the disease first hit the U.S., is one of our partners. And then we're also delighted that Meg McGrath, and Meg just won an award. She's quiet and humble, but she does really dynamic work down at Long Island Experiment Station. She, from a pathology perspective, is also looking at the chemical control agents and strategies. And in concert with Robert Pine, my grad student, and Andy and myself, where we're doing the genetics and breeding of the plant itself, not as interested in the pathology of the downy mildew. We've done some studies and some dialogue crossing that we'll be reporting on later in concert with Meg at her station out on Long Island. So we're pleased that Cornell University, UMass, and University of Florida have joined with us at Rutgers. At Rutgers, it's Andy who just introduced me. He's our state, many of you know him, but those out of state, he's our state plant pathologist and excellent vegetable crops extension specialist and researcher for pathology. And he's taking charge and leading in concert with our grad students, Catherine Homer with Catherine? Somewhere back there? She's also quiet, but just finished a master's in looking at the different control agents that work effectively in the New Jersey environment. And she's reported this at the prior years in New Jersey. And she's assisted, and she's also full-time at IR4 and assisted with Bill Barney. Bill, raise your hand. Okay. And looking at, again, the chemical control from the plant pathological levels, and also working with the companies trying to get new approved compounds registered for you as growers. I know as growers we don't talk about it, of what you can use that isn't registered versus what you could use that is registered. But from our perspective, we're trying to make sure that everything you can use is properly approved and registered, so everything falls within legal guidelines. And then Bill Sharappa and Rick Van Rankin and Wes Klein and Pete Nitchie and others from our Cooperative Extension Service here at Rutgers have done a lot to translate a lot of this information that we're finding out to you as growers. So that's our research team as a whole. And we came together. Funding is not easy, you know. Professors never have enough money, right? We always tell you we're broke. Universities are always broke, everybody's broke. And so, but we didn't, what we have is tenacity. We may not be that smart, but we have a lot of tenacity. And I'll tell you, it took us a couple years and a couple rejections and non-acceptance of that, the denial of the rejection. And I think they just gave us the grant because they didn't want to hear from us anymore. But USDA gave us a lot of money to bring together objectives in the 4 following areas. Okay, the first is to identify sources of resistance, that is genetic resistance of plants, and then, if identified, to develop improved germplasm of varieties. You know, varieties might take 10, 12 years, but at least we could identify, improve the germplasm, look to see if there are varieties that we could create and make that have tolerance to downy mildew, Fusarium, wilt, and shilling injury. And later today you'll hear there's some interesting new news about Fusarium wilt, okay? And relative to the lines that had been theoretically resistant to Fusarium wilt, the isolates that are now being picked up, being screened to those same Fusarium-resistant plants, those plants don't show Fusarium resistance anymore, and shilling injury. And then to develop standard laboratory techniques that would be used for the detection of Fusarium wilt and downy mildew, that's largely led by UMass. Develop improved management strategies to effectively control downy mildew, because there's usually not any one answer. Each of you are basil growers, you all grow basil differently. Alan, you grow it very differently than somebody in a different state. And your neighbor will grow it differently than you. And there may not be one way or one wrong way to grow it, but you're using different ways, and you use a combination of arsenals or tools. And the objective really of our consortium was not to necessarily provide one answer, but using a number of different strategies to try to make the downy mildew problem manageable and be able to mitigate against it. So develop improved management strategies to effectively control the downy mildew based on disease forecasting, monitoring system. There was a beautiful cucurbit monitoring system that Meg put in together for cucurbit diseases, for downy mildew, and she expanded that to include basil, and she'll talk about later this morning. And through the identification of effective organic and conventional fungicides, and we'll see where we are and what we haven't done, but a number of years ago, before this consortium started, we had fewer registered pesticides. So to even be able to get so many compounds out there approved for use was a really successful approach as far as we're concerned. And then develop a cost-benefit considerations, and I guess I forgot the 2 others in our program, Ramu Govindasamy and Surendran. You can't always forget the person who deals with the money issue, but somehow we forgot Ramu and Suren that do the budgetary analysis in concert with Bill and others. So look to see if, you know, what does it take? How much are you willing to spend to buy improved seeds that are genetically tolerant, or I guess as Enzo, you talk about intermediate resistance, and or are resistant to downy mildew seeds, which will be costly, and or even the chemical control agents that a lot of growers don't always necessarily take into account, even though they're doing weekly sprays in the course of their production of basil. But we have seen the last couple of years since we started, there's many people that no longer grow basil, even in New Jersey and other states. The risks were too high, the costs were too high, and they just said, why bother? We're going to drop off not growing basil. So we know that downy mildew is an extremely destructive disease in the United States. We know there's no commercially available sweet basil with resistance. Okay, now I say with tolerance. There does look to be some germplasm out there that shows some tolerance in some areas under some conditions, which is exciting, which is great. And we'll hear more of that through the grower panels later this morning and through our presentation in the afternoon on the genetics and the tolerant lines that have been created already. The lower left is just a fancy picture to let you know we're scientists, so it shows you the downy mildew coming in out of the stomata. But more than that, it really shows you really what the disease looks like actually on the leaf itself, with a resistant plant to the left and a susceptible, resistant plant to the right, above on the upper right, resistant to the left, and actually a resistant sweet basil plant that Rutgers has developed that has true resistance on the bottom part of the left-hand side. This is a typical type of basil that a variety that many of you grow, De Genova, 33 days after planting. I'm going to stop here because right now the welcome is going to move into what we're now doing according to the different approaches. So let me suggest that at the beginning of the workshop, we're going to be talking about disease forecasting and monitoring and where the downy mildew shows up, when and how. We're going to move into the different chemical control agents, that is what's available for you now, given the varieties that you have that work and not work. And we have a lot of different studies from each of the different states going on that. Then we're going to have a grower panel that will have, I guess, some seed company representatives that will talk about, you know, what their observations are, what varieties might be working for them, or what techniques that they're willing to share work. And then we'll break for lunch in the afternoon, and then we'll talk about the genetics of it. And when we talk about the genetics, we'll show you the studies we're doing here as a quick, where our goal is really to develop what we call these immortal, or these inbred lines from which advanced populations genetics can occur. We're looking at the genome of it. You know, when we first started, we had resistance actually, but resistance in really funky basils that didn't have the right smell. So we use these kind of flavor and aroma charts. We do the chemistry of aroma and the chemistry of aroma taste. We put them in different, and this is just a highlight of what you'll be seeing later this afternoon. Is Robert Pine yet here? Okay, Rob, okay. So this is just a highlight of some of the work, of his leading breeding work that you see in the afternoon. But this is what we hope will captivate you to stay in the audience all day long, because you're going to see really what happens when you cross resistance with susceptible plants. Some of the progeny that comes from that. Some will be acceptable, some not acceptable. Some even acceptable in a look. It may not be acceptable in aroma. And then what are we doing to try to create that correct aroma with plants that are actually resistant, or some of the plants are not resistant, but they have a high degree of tolerance. Tolerance is a continuum, and we'll talk about that and how we define tolerance or intermediate resistance, because where along the gradient it works. But these are sweet basils in a field where everything else has crashed due to downy mildew. So we feel confident when we look at this, we're getting the phenotype, and in some of the lines, it's really looking good. So we'll give you the detail in order where we are and what we have and what we don't have, by the way, as we're moving ahead. And we'll talk briefly about some of the progress we've made even on the chilling tolerance of the screening of basil. And you can see, I think all of us would rather grow the one on the right than on the left. Okay? The data and the stats doesn't really mean anything. You can kind of see it. The picture's worth a lot. And we're very happy with the plant on the right side versus the plant on the left side. So with that, there's Dennis. I couldn't have a slide without a picture of you in the back, okay? I couldn't put a picture of it because Dennis Del Ponte and Mr. Mint and his boys, his family, have been really instrumental in working with us and doing a lot of collaborative trials at his farm, and we really thank you, Dennis, and your family for that. Okay, so without further ado, let me just ask, before I turn over to the second speaker, how many growers do we have in the audience? Okay, this is good news. And how many seed companies do we have in the audience? Okay, what seed companies are out there? I'm getting too old, I can't see you that far back. So stand up and tell us what seed company you're in. Is that Johnny's, I'm assuming? Yeah. Okay. Dominic, you going to stand up too or not? Okay. Any other seed companies? Any other buyers or distributors of herbs? Okay. So the whole point of this workshop, it's not just only a series, we hope, of lectures and presentations, but there'll be some opportunities for dialogue, exchange, agreements, disagreements, to see how we could move everything forward. So with that, Andy, are you introducing the next speaker, or am I? I am.